Hello students, welcome back. Hope you have already watched the previous two videos. Right now we are going to learn dosage compensation. Otherwise it is called Barr body. So let's get started. As you all know, in humans we have XX and XY type of sex determination, which means females will have two X chromosome and males will have only one X chromosome and one Y chromosome. Since males produce dissimilar gametes and dissimilar chromosomes, we call them as heterogametic males. Since female always produce same type of chromosomes in their eggs, we call them as homogamatic homomorphic, which you already know now. Now let's talk about what is dosage compensation or Barr body. So this is related to the X chromosome. So that we will be seeing in another slide. So if you look at this picture, you can see this is a female individual progeny and this is a male progeny, because based on the type of sex chromosome we can understand which is which, right? Now, so let's talk about the X chromosome first. So this X chromosome does not participate only in the sex determination. It also participates in lot of vital body functions and mechanisms. So imagine in males we have only one X chromosome. In the previous video when I taught you guys about Y chromosome, I told you the sex chromosomes are not homologous because they don't contain the same genes. Say for example, if you take male chromosomes are called hemizygous because you don't see same type of gene completely in both sex chromosome that has been inherited by the progeny from their parents. Say for example, if you look at this X chromosome in males, you have 5% of same genes will be available or present on Y chromosomes. So we call them as pseudo autosomal region, which means the same genes that is present in the X chromosome will be present here only for 5%. The remaining genes that is present in X chromosome will not be there in the Y chromosome. So that region, what did we call? We call that as non-combining region of the Y, and this is where we have this male sex determining genes also. Based on, like, in that non-combining region we have functional genes as well as non-functional genes. In the functional genes we have the sex determining Y gene, which is responsible for the expression of males. So this we know now. So as I told you, X chromosome does not participate only in the determination of sex. It also participates in the lot of vital body mechanisms and body functions. So it has been reported that the X chromosome, especially the sex determining X chromosome, contains as many as 1,000 genes. So in males, so we have one X chromosome and one Y chromosome, which means in the X chromosome you might have 1,000 genes, while in the female we have two X chromosomes. So what does it mean? It means we'll have double the time genes when compared to male and male humans, because in female we have two X chromosomes, right? So if we have two X chromosomes, say for example, these 1,000 genes are responsible for producing various genes, and all those genes produce lot of different proteins that code for different functions in the body altogether. So in males we have 1,000 genes and that will produce different various protein and perform functions, while in the female, since we have two X chromosome, the number of genes will be or will also be increased, and the proteins that are produced in these two X chromosome also will be double the time when compared to male animals. So this will result into incompatibility between two sexes, because when you take any animal, both animals should contain same type of counterparts, right? Same type of mechanism and same type of this chromosomes and cell structure and everything. But since it has two X chromosome, it produces too much of, it might produce too much of dosage that includes proteins that might trait or code for different various function. So then there will then the female as well as male animals will not have equal or compatibility in between two sexes. To avoid that, what the female progeny will do during the embryonic development, one of the X chromosome will be inactivated. Okay, so this process is called X inactivation or dosage compensation. So I hope you all understood up to this. So this dosage compensation or X inactivation. So how it will be inactivated? So if you look at this picture, if you look at the picture, you can see here one X chromosome is being activated and one X chromosome has been inactivated. So how will it inactivate? The another X chromosome will condense and change into thick chromatin tightly compact material. Okay, so that we call that as heterochromatin or chromatin. So this was first identified by Barr in 1949. So in cats, when he observed in the cat's nerves cells, he could able to see among the autosomes, the sex chromosome, in the female sex chromosome, one of the X chromosome looked like a condensed material. So he called that condensed material as Barr body. Since he has discovered that, his name has been given to that discovery. So this Barr body again is responsible for the inactivation of the X chromosome to compensate the dosage incompatibility was suggested by Leon, hence her name has been given to it. It is also called Leonization, otherwise people say that as Leon's hypothesis. So I hope you all understood up to this now. Now, so you must be having a doubt. So when a female inherit one X chromosome from the mother and one from the father, so they will contain two X chromosome, right? So you must be having a doubt which X chromosome will be inactivated, whether the X chromosome inherited from the father or mother will be inactivated. You must be having this doubt. So what happens during, as I told you, this X inactivation will be carried out during the embryonic development. So after mitosis, what happens? After fertilization, cells will be continuously dividing. So in during the embryonic development, say for example this cell, so in this cell the X chromosome inherited from the mother will be condensed, which means it will be inactivated. Likewise in this cell the X chromosome inactivated from the father will be condensed and inactivated and it will look like a Barr body, right? So likewise alternatively one from the mother and one from the father will be inactivated in different cells during the embryonic development. So this was first identified or discovered by observing these calico cats. So if you look at this cat doesn't have only one particular color, fur color. It has mixed color, one black as well as one brown color. Say for example, it has inherited one black color from the father and brown color from the mother. So during embryonic development, this X inactivation would have been conducted or carried out. So at that time, say for example from the father it has inherited black. So these cells which are present in this particular region would have this father X chromosome. So and the mother X chromosome would have been inactivated here. That is why it has only black patch here. Likewise, here the black colored X chromosome which has been inherited from the mother would have been inactivated. Hence, the brown color inherited has been expressed. So based on that, they came to know this kind of genes are called sex linked genes. Okay, sex linked genes. So because since this has been expressed due to the sex determining chromosomes, they call that as sex linked genes. So when we know that we have different types of sex determination, right? XX and XY, humans and fruit flies will come under that category. And I also told you XX and XO sex determination is also there. In that, you cannot find bare body in female animals, because since females have only one X chromosome, you cannot find bare body, while you can find bare body in males. Likewise, due to chromosomal disorder in humans, sometimes male progeny will inherit two X chromosomes from the mother, so that particular male progeny or offspring will have one bare body in their every cells, especially on the place of sex chromosomes. Now let's talk about haplodiploidy in honeybees. So we have already studied this parthenogenesis during our first chapter. I hope you all remember that. So when you talk about this honeybees, unlike other animal, they will not contain any sex determining chromosomes. Instead, the number of sets of chromosome determines the sex of honeybees. Say for example, if a queen, it has not undergone any mating process with a male, so what happens, there won't be any fertilization, and the eggs that are released by the queen will always contain males. But so hence we call that as haploid. One number of chromosome only is there, which means one set of chromosome that has been inherited from the mother alone is there. Hence we get every progeny that has been inherited without fertilization, we get male workers. Otherwise we can call that as drones, right? So if you take after mating, say for example this female has undergone mating process, so it will result in the formation of worker bees and also queen bees. So they will contain two numbers of chromosome, which mean half the chromosome from the mother and half the chromosome from the father, both will be inherited. So they will be fertilized and will contain bees, worker bees, female worker bees, and queens will be produced during this time. Since haploid as well as diploid type of sex determination is being taken care in this honeybees, we call that as haplodiploidy in honeybees. I hope you all understood, and I'll see you guys in another video. So learn up to this. See you guys in the regular online class. Take care. Bye bye.